So the benefits of dried plasma spots have just been nicely reviewed, and the workbook that you have has a whole lot of material in there that I've removed from this, so the workbook will not follow this exactly. I'd like to get my pointer well behaved here. So I have my employer.
So I put a mouse here, which might be a little easier for you. And if you want
Oh, this
just to-
works on great.
And if you want to advance slides, just use the arrows.
Good.
Or this here. But when it's time to click on the video,
Yep.
you can just use the mouse.
I have a history long ago in anti-doping, but I am not currently in anti-doping, so a little bit of transparency here that my employer is a regulated bioanalytical laboratory. We do LC-MS all the time for the pharmaceutical industry, which is where some of these drugs come from in the first place, but they're used for anti-doping. So I want to talk about an alternative matrix, which is plasma, and I would remind you or point out that plasma is the favorite matrix in the pharmaceutical industry for quantitative measurement of drugs. It's always has been done for years in plasma. Why? It's easier, I would suggest. And what happens if you take a tube of blood and spin it with a centrifuge, you get this yellow liquid on top. And that requires a centrifuge, it requires a phlebotomist, it requires a lot of things that are not convenient on site, if you will, for sample preparation. So the takeaway message today in my presentation is that we're going to consider an alternative to this, and that alternative looks like this. That wetness that you see on a piece of paper, cellulose, pure cellulose, is plasma. It is created by putting a drop of blood onto a card that looks like this. And the red blood cells, which are a few microns, very small particles, are filtered out. So the plasma is created by filtration rather than centrifugation. You don't need power, electricity, and so forth. And so that's the take-home message. And the device that I'm going to describe is a work in progress. It's not commercially available. We did have a micro grant for the last 6 months, which was to generate a prototype which hopefully could be manufactured by someone. And that's to be done in the future. So this device is not available as yet. So I'm going to talk about these two matrices. I will say very little about DBS because you've just heard a nice presentation of that. We've done a lot of work with DBS, dried blood spot analysis. I might add that we have Q Squared Solutions has a laboratory in Ithaca, New York, where I am, and also in Indianapolis, and we do thousands of cards a year. Almost tens of thousands of cards for the pharmaceutical industry. The biologists that collect blood from a tail prick of a mouse, that's how early drug discovery work is done with that. And the biologists love this concept of microsampling. It's very easy to do. We can turn around the results in a day, basically. They show up in the morning, we give them results at the end of the day because it's so quick and easy. I'll discuss the technology needed for testing these alternative microsamples. That technology exists. It's not new technology. It's what we can purchase. Dr. Camber showed one example of those. There are benefits and challenges. No technique or no approach is perfect. We have a toolbox of tools that we use, and this could be, I think should be, one in your toolbox for the future to consider because of the apparent benefits, but it's not a perfect world. Drugs distribute throughout our body, to the tissues, to the blood, to the plasma, to the breath, to the tears, all over. And unless we take the whole body, the whole person, we aren't going to get all the drugs in one sample. And as you've just heard in that discussion, plasma covers a lot of drugs, usually the parent drugs, the unmetabolized drug. Predominantly, metabolites are more likely to be found in the urine. So the benefits, the representative applications, we have a lot of them, but I'll just show one or two that are relevant to anti-doping. And I'll close with suggesting that, or showing you that there are other commercial devices in the works on the market that can be available to do similar things. So the challenges that a laboratory has, I might add, just as Marilyn said, I'm a proud board member of SMERTL, and that's a disclaimer that basically that's my current link to anti-doping as being on that board. And so I have some experience in the prior years of what a laboratory has to deal with. Athlete cooperation and objections to being sampled. We just saw that with the dried blood spot, the athletes much prefer a finger prick to a needle stick in their arm and be a witness while they collect urine and so forth. Collection of sufficient sample. I might add that yes, mass spectrometers have become very, very sensitive, but this microsampling involves from 1 to perhaps 5 or 10 drops of blood. The device that I'll describe here provides 40 to 50 microliters of plasma. That's actually plenty by today's modern techniques of LC-MS. There's exceptions. Sometimes drugs are very, very low. If it's 1 to 5 picograms per ml, we actually publish 10 picograms per ml by dried blood spots on 10 microliters of blood, but that's the exception. Most drugs are readily obtained or detected quantitatively, and I want to distinguish, Marilyn just mentioned qualitative analysis. That's what is this drug? But most of the time, it's more than that. It's how much drug is there, how much is allowed, and that's quantitative analysis, and that's a whole lot more work. We need internal standards. There are other issues associated with it. So the collection of these samples is important. The ease and cost of collecting the sample, I come to learn, and it's not hard to realize if you pay your FedEx bill for shipping dry ice and samples all over the place, it's very expensive to ship these samples that we're now doing in vacuum container tubes, as well as the ice, the keeping it cold and so forth. Maintenance of chain of custody, very, very important that there's nothing tampered with in the sample. Maintenance of sample security en route in that sample transportation. The cost of those shipping samples, as has been mentioned. Not mentioned too much is the stability of the drug. Actually, in many cases, not all, we have some exceptions. Darn it, there's always exceptions to rules. We thought, and it's reported in literature, that when you have a dried plasma spot or a dried blood spot, the enzymes are now dry, they're not active like in our bodies, and that a dried blood spot would be more stable, the drug being more stable, not decomposing on the card. That is generally true, but there are some exceptions. We have had to bathe or place the dried plasma spot card in a tamper-proof bag with nitrogen or argon or cool it. Now those are exceptions, but the majority of the drugs are more stable in shipment. They don't decompose in shipping, and that's another benefit, but it's not a universal benefit. The laboratory challenges handling different types and volumes of samples. 25 microliters is one sample volume. 3 or 4 milliliters is a whole lot more. And then in the past, we used to extract and work with milliliters of sample as opposed to what we're now doing now. Most bioanalytical methods now use 10, 50, 100 microliters is plenty of sample, driven by our ability of getting better at this, but most importantly, the sensitivity improvements in commercial systems, analytical systems. And of course, long-term storage for possible reanalysis. And laboratory harmonization, consistency in techniques used. I think that was just mentioned in the discussion. So I don't have to review too much of this. I've tried to, through this presentation, highlight in red blood, because it is blood, is red, and plasma being a yellow, if you will, color. We've heard about oral fluids and breath and other things. These are various fluids that are available, and I'm going to focus on plasma. Drugs and their metabolites can be found in 1 or more of these. So this is what most people don't like to do. Some people really don't like to do it, and athletes would rather not have this, and they can be more accepting of a finger prick. So this is a lance, a spring-loaded lance, if you will. You touch it to your finger after wiping it with isopropanol, and a little bit on the side, not where the callus is. It's really not hard to do. Many people do home tests now with dried blood spots, as well as other kinds of tests where the bloods can be sampled with a home device. So capillary blood. I might have mentioned too, it hasn't been mentioned that venous blood is what we have here. This is more like capillary blood, and there's some discussion and argument, are they the same blood? Well, they came from the same person, the same body, and generally that we believe, without extensive studies of all possible drugs, that they are the same blood, but there are some issues that are being discussed and more work needs to be done there. So microsampling is shown here. This is applying blood to a card like you just heard described, and we're going to convert that blood by filtration to a plasma spot. Hopefully you can see there's a wetness there. It looks like it's wet, basically. And that's the plasma that can be produced onto a cellulose, think paper, pure paper that the CDC controls, very pure paper, so there's no other drugs, no other contaminants on it. You can collect 3 or 4 samples. You can be the B or C sample, or they can be stored for later. and so forth. So we're talking about a dried plasma spot card. The benefits, I won't dwell on these because they've been nicely reviewed prior to me, but this single finger prick here is the key. And the key in the workflow in doing this is to not smash the finger, if you will, press it on. It's important that the drop hang from the finger and the drop touches the paper and it wicks into the paper, the cellulose, without your finger touching it. And that's an important step that's not hard to learn. It's also helpful to perhaps point out that we don't all bleed the same. I happen to be a more difficult bleeder. We've done a number of experiments. I'm the healthy volunteer in some of these experiments. And there's a little bit of a knack, but it's nothing people can't do. Having your hand down like this, warming the hand, and having a procedure that it's quite easy to get blood from almost everyone. And so that previous slide shows the idea of a drop hanging from the finger, touching the paper and wicking into the paper. It works very, very nicely. And that's usually in the neighborhood of 10 or 20 to 30 microliters. Usually a drop is about 20 microliters, and this card can accommodate up to 30 or 40 microliters. I might add that since the 60s, the Guthrie card has been used. Every newborn baby in the developed world, not only US and Europe, but not in the middle of the jungle, but in the developed world since the 60s, has been sampled with a heel prick, shown right here. And the heel prick is, the blood is put onto a paper, on a dried blood spot card, and it's sent to a major laboratory in New York State. Is Albany. Albany, New York has a state laboratory there, and the last count I knew they had 10 triple quadrupole mass spectrometers running all day long, 24/7, analyzing these results. And so a newborn baby in the hospital is analyzed, the blood is analyzed for inborn errors of inborn errors of metabolism, mainly a series of endogenous biomarkers, basically. And if a baby has a problem and it's not caught, they will likely die in a few years, 1 to 3 years. If it's caught simply by a diet, they will live a perfectly normal lifetime. It's a real success story for this kind of technology. It's also done by mass spectrometry. It's a success story for mass spectrometry. I might add it's not LC-MS, it's flow injection MS. Chromatography does a great job, but it takes time, and with so many samples, they run lots of these by flow injection, just flow an injection of the sample into the device. So this is a really well-established technology, and perhaps it's time to get this kind of technology into anti-doping. So this is what I'll be describing. A card that has plasma, dried plasma on a round surface of plasma, and we're going to elute that card online. As Dr. Camber showed, that automated system from Spark Holland, which is a company in the Netherlands, that can do that, or it can be punched. The manual approach is to use a punch to make a disc, if you will, cut that out and extract it. Can be done either way. And so, reminder, plasma can be produced by centrifugation, or it can be produced by putting blood onto a device such as this and let the filtration take place. So here is the current embodiment of this card. Thankfully, to the microgrant that we had, we were able to bring this to the current position. The color, don't worry about the color. This is 3D printing. 3D printing is good for prototyping. It's not good for mass production. It's slow. But you can have a CAD drawing. We worked with a colleague and friend to do this, and so this is 3D printing, and the color can be different, and don't worry about that. So the finger prick is added to this in this way onto this spot, and then the blood, I got a cable here that's not behaving. The blood and plasma spots, this is I'll show a video in a moment, but we call it a book-type device because it can be opened like a book. So there's the spots, and we open it like this and expose the blood and the plasma. We bring it all the way around, and that's the way it's basically analyzed. You'll see that in the video in a moment. And so here it is being opened up and also put into a tamper-proof device. I want to thank Daniel Eichner for pointing out to me that we needed to make this tamper-proof and tamper evidence, if you will, and I'll show that too. And so in this example, it's placed into a plastic box, if you will, and sealed, and then with tape on it outside like this, so tamper-proof tape. Alternatively, as shown earlier, it could be put into a bag. There are various tamper-proof bags that the police use and so forth, so that's work in progress to see how best to do that. So how does that filtration take place? How is the plasma really separated? Right in here, hard to see in this example, is a paper-thin membrane. If you've ever seen a transparency for a photocopy or something, it looks like that, but it's not that. It's that thin, it's transparent, but it goes right in there, and that is filtering out the red blood cells. And if we look at a scanning electron micrograph, this is that thickness, that paper-thin thickness expanded, and these little red specks are red blood cells. So the red blood cells are being restricted in that. Plasma is seeping through onto the paper, the cellulose that's on the bottom. Now, and Dr. Camper talked about blood spots. There is an elephant in the room, if you will, or an issue with the quantitation of drugs in dried blood spots. We know it, and most everyone else knows it, and it has to do with what's called hematocrit, the thickness of the blood. Most of us that are healthy and normal, our hematocrit's around 45 to 50%. And that means that there's a certain percentage of plasma in that blood and the rest being red blood cells. If a person, an athlete, has a very low hematocrit, 30%, there's a lot more plasma present. It's more fluid. And when you put that on paper, imagine spilling some red wine on a tablecloth. It spreads like that. It's dispersion. And a thick blood will not disperse as much as a thin blood. So a thick blood is up here at 60%, and that makes a smaller spot. Can you see that that spot is smaller because there's a lot more red blood cells? In this case, the thinner blood with more plasma in it spreads more. So what happens when we take a punch, a fixed diameter disc? We have different amounts of drug because of that size of that spot. You're going to have a lot, and let me show this picture over here. Here's 30% going up to 60%. This is a little bit of an optical illusion, but those white spots are all the same diameter, but they are removing a very different amount of drug from that, and that really messes up quantitation. That's the problem with dried blood spots. And the key is to put a known volume of blood. Like with a pipette, that's not convenient in the anti-doping world. There are some devices in the works, and Dr. Camber showed one, and I'll show one later, that it can actually deliver a known volume. If you do have a known volume of blood, not just a hanging drop, we don't know what that volume is. We don't want to have to know what that volume is, but if we have a known volume, then if we extract the whole spot, it won't matter what the hematocrit is. I know this is kind of confusing, but it's an issue with dried blood spots, and I'm here to show you that we don't have that issue with dried plasma spots. So each extraction site above the white spot represents a different quantity of sample. So what about hematocrit effect on DPS, dried plasma spots? Not a problem. Plasma is plasma. After it's been filtered and it's away from the blood cells, it spreads the same. And so we don't have to have an issue. We're going to have the same aliquot, same portion in our punch or our elution spot. And that's one nice benefit, if you will. Okay, so here's a picture similar with what was shown before. We have this plasma card. We flip it over and put it into this robot. And we're eluting it through with solvent, goes to the on through this robotic system and onto the mass spectrometer, and we get quantitative results. So here's just one example, dried plasma spot analysis of 9 opiates and stimulants. There's the structures, there's what it looks like on a card. We're eluting one of those spots. We can go back later and do the next spot, or it can be saved and analyzed later. And so here are, in 1.5 minutes to in about a minute, you're getting 9 of these compounds separated and quantified, each with a stabilized slope internal standard. So we applied blood, we got plasma, and there's our results in this number. So that's the story of the plasma. But remember, this book-type device has another layer that's got blood cells on it. Can we do something with that? Well, several months ago, I sent to Smyrtle, Dr. Daniel Eichner, a dozen or so of these cards. They were prototype cards earlier than this, to take a look at the blood layer. So this card now gives you two aliquots from the same sample. You can look at the blood, the red blood cells for hemoglobin, for proteins and so forth, and you can look at the plasma for drugs and metabolites if they're present. And so here then is what Dr. Eichner, along with Dr. Holly Cox, looked at, and I'm happy to say that they came through for me with a hustle towards the end and produced the results which I'm about to show you. So these are slides from Smyrtle. and Dr. Holly Cox and Daniel Eichner produced these, where they're looking into counting the red blood cells on this separation. I might add that this is called an IPOC filter. That's International Point of Care. It's a company that makes these things. They're available in Toronto, Canada, and couple of other places. So that's this upper layer where the blood cells stayed on top. We're going to analyze that blood for different things. So here's adapting the blood passport to dried blood spots, where an athlete, and this is what this card looked like in the early days. We held it together with paper clips. It's a little more sophisticated now. Paper clips are inexpensive, by the way, but to keep the membrane close to the cellulose so the plasma would seep through, we use paper clips. And so that's what they're doing, is applying blood to this, separating out the red blood cells and analyzing that upper layer, setting aside the plasma. So the blood samples are shown here, and in that we have the red blood cells, the immature reticulites, the white blood cells, the platelets. These are blood proteins, and a longitudinal profile is monitoring an athlete over a period of time and seeing how those measurements can be recorded, and whether that person is using transfusions, what the hemoglobin is, the various other things that athletes do to dope their blood in other ways. And so the workflow shown here, here's the upper layer, the red blood cells. In this case, I believe they punch that out, and you wash to remove the soluble proteins, and the red spot becomes a white spot, but there's still all these things in there that were not washed away with water. And then these are digested, enzymatic digestion, that's what the scissors are showing. The proteins are digested into surrogate peptides, which are measured with LC-MS on the high-resolution Orbitrap mass spectrometer. And so hemoglobin measurement often can be done colorimetrically, and it's quite linear. The takeaway message here, this works pretty well. This is analogous or similar to other ways without large volumes and lots of extra work that's done. It's necessary to do these analyses. So here then is with different hematocrit. I mentioned hematocrit from 30, here it goes to 50%. Basically, they group together as they should. Matrix issues are not a problem. Apparently, for these analyses. The red blood cells are linear and so forth. So the analysis of the upper red blood cell layer looks favorable. Recovery, recover the drug, that's always an important one. The historical approach is the Whatman surface, and that you can see in each case is slightly better, but the IPOC membrane, this polymer membrane where the red blood cells are on this card, are quite competitive, quite comparable, satisfactory, if you will. What else is available commercially? Just very quickly, for micro devices. This is the device called Advanced 100. It's lateral flow. The blood's applied to, if you will, a piece of filter, a paper. It migrates, and there's the plasma. The York test device has been out for some time. It only produces 2.5 microliters of plasma. That's really too small an amount. We want 20 or 30 microliters. So we've looked at this, but this is impractical, I think, in your world. And then there's this device by DBS Systems that I think Dr. Camper showed. This delivers a known volume of blood, which would be good for dried blood spots. And then there's this device by Neoterix, which spun off from a company in California, Phenomenix, and this is rather interesting too. So here's what this little device looks like. You apply blood to it, and it comes, your blood goes from your fingertip to here, you close this like a book, and that brings this surface in touch with the paper, and you get a dried blood spot. So that's a known volume. That's a good thing. That's not making plasma. Then in this device, it's a tip that looks like this. It's white, and you touch the blood on your finger, and it makes it wicks up into that device, becomes all red, of course. This does not produce plasma either. I wish it had a device to do that. I've learned that neonatal, or babies, young children that don't like to be pricked, they like this because they like to see the thing get red. They're amused to see that, whatever it takes to have a kid give a blood sample. So this is a very interesting device. It's commercially available. And then there's this one, MDI Rapid Plasma Separator. I don't care for this because you have to have a manual device. You have to pipette from a reservoir the plasma, but it does produce plasma, similar to what I just described. And then there's this one from Texas, a Hemospot. This looks like a star, or a little, like a star like that, and you place blood in the middle of it, and it radiates out, and you tear these off and do various tests. That's kind of impractical, too. It's kind of a cute idea, but you could tear it off and put it in a multi-well plate and extract it, but so be it. That's another device that's commercially available. So finally, summary and conclusions, opportunities and benefits from a DPI device. Remember, there's pluses, but there's negatives. less invasive finger prick, blood sample separated into red blood cells, superior sample stability in many cases, not all, simplified, cheaper way of shipping, lower contamination risk, biosafety. That's actually handling blood is usually done, needs to be done carefully. Collection of accurate blood volume is not necessary for plasma. No hematocrit issues with DBS. And the technology and methods exist right now to do these if the device is available. Challenges, expanded applications to validate. We've looked at only a handful of classes of drugs. There's many more drugs. We've not done anything with steroids yet and other kinds of compounds. Acceptance of collecting microsamples. A lot of labs still like to have more, just they're not used to scaling down. I think they need to get with it because the trend is towards smaller samples. And there is the manufacturing source for this new device is not yet available, but we're in touch with some folks to do that. I want to acknowledge then the PCC for their generous support of our work. Shimadzu consigned the mass spectrometer, which we eventually purchased, I might add, for over a year. And Spark Holland has consigned the automated dried plasma spot elution device that I and Dr. Kemper have shown. And last but not least, Dr. Imelda Rayana is the postdoc who is here with us today, who did the work I've just described. Our prior postdoc, where we published work in late 2016 on dried blood spots, was Dr. Ruth Verpletz from Belgium, and she's gone back to Belgium. So I'm having great fun having industrial postdocs. I cannot tell them it's Cornell like I used to, but I can give them a job afterwards, and some of them have stayed on to be employees. I thank them, and I thank you very much for your attention. Thank you.